Fantastic. It's doing something. You kind of got the introductory, or most of the introductory bits. So my day job is in the University of Southampton, which is why their logo is there. But that's nothing to do with botany, so let's not go there. A lot of my holiday time gets taken up with the Outer Hebrides. So I live in Southampton and record in the Outer Hebrides, obviously. So let's not do that either. But you will get quite a lot of examples of the kinds of fungi that grow on the kinds of plants that occur in the Outer Hebrides, because that's where I go a lot. And I'm also part of the Welsh Parasitic Microfungus group, but more about them in a bit, so I won't say too much. The last bit, the logo at the top. Today is UK Fungus Day. You all knew that, didn't you? So I'm afraid you have to hear about fungi as well as plants for a bit. But I hope it'll be interesting. So I'm going to tell you a little bit about my story with plants and a little bit about what I've done. So we'll think about getting started with plant parasitic microfungi, and if you're lucky or unlucky or something, you might get hooked on plant parasitic microfungi, and I kind of got there a bit. We'll talk about Wales, because a lot of activity has been happening in Wales over the last decade or so, let's say, including some red lists, some ID guides. Then I'll run you through the different groups of plant parasitic microfungi that there are out there. You'll see some of those as we go along as well, but we'll at least give you an idea about the different types of fungi that you might find if you go looking. I got quite interested, and you get a bit tied up in taxonomy, the same as you do with plants, as soon as you get into something a bit deep. But I'll talk just a little bit about species and nomenclature and what that means for what you might do in the field and what you can do. And then I'll give you a bit of my story about how I got a bit more than hooked and got quite excited about stuff, and some conclusions at the end. So let's think about getting started. Most people get started with rust fungi. And you've already got one huge advantage. You know what the plants are. Once you know what the plants are, it's relatively straightforward, because most of the plant parasitic microfungi are species specific or species limited. Sometimes it's a small group of species. But if you know what host your fungus is growing on when you start, they're actually much easier than all those horrible mushroom-shaped things, where you have to go cutting sections and looking for clamp connections and cystidium. Don't go there. I don't do mushrooms. So if you know your plants, you're already halfway there. And then you need somebody, which could be me this weekend, if you like, saying, find me a rust. In my case, it was Arthur Chater, who some of you will know very well. And we were out reasonably regularly walking, and he would say, find me a rust, find me a rust. So you look, oh, what's this? Is that a rust? No. Is that a rust? Well, yeah, that's a good one. I'll have another one. Find me another rust. And after you've done that half a dozen times, you think, oh, I wonder what that is. That's really quite interesting. Can I find another one? And somebody already mentioned to me Debbie Evans from North Wales, and she's you end up with this tick list in your brain that says it's one of those. Oh, that should have a rust on it. So has it got a rust on it? I found whatever it is, Epilobium hirsutum. Has it got a rust on it? So apparently Debbie says that all the time and winds people up. So here's a really common rust. You won't find it at this time of year. It's one that's out there in the spring, but this is Puccinia urticata. It has Esia, which is one of the rust stages, more on life cycles in a moment, but it forms these fantastic galls like the ones over there that cause the plant to swell at the same time. But this is a rust you can find in the spring, and it alternates with sedges. More about alternation in a minute as well. Rusts are common. It's practical to go for a walk and find several to many rusts. So while we were out this morning with our app, about 10 rusts on my tick list. There's one right outside the door, so you can go and find that one later if you want. The trick with rusts is to walk slowly, because these things are generally quite small, although you might get some indication. The leaves are often discoloured or look a bit odd. So it might be a hands and knees job if you're really down in the herb layer and it's a bit mown or rabbit bitten or something. But upright's okay for taller plants and trees and shrubs. you end up turning leaves over, especially the manky looking leaves. So if you're out there for botanizing, you want something in perfect condition. If you want rusts, you want something that looks a bit manky. There's Puccinia coronata, which is a really common rust on lots of grasses. That one's on Yorkshire fog, and it's quite often in epidemic proportions at this time of year. So you can go out there and find piles and piles and piles of it. Rust fungi have really weird life cycles. They have 5 different spore stages. So let's start over there where it says naught, which is the first ones. So the first infection you get on a plant is a set of, usually a bit inconspicuous actually, some things called pycnia or spermatia, and you have to struggle sometimes to find those. We won't go into the technical definitions. I can if you really want to ask me later. So those pycnia create pycnia spores, which germinate to create ecia, and those things that we just saw on the nettle were ecia. So they're stage 1. If there's alternation, it's usually those stage 1 spores that go to another plant entirely, and they create stage 2, logically enough, called Uridinia, and those are the kind of brown powdery things or orangey powdery things that you see. And those, they can cycle, so those Uridinia spores can make more Uridinia during the year. As we get towards the end of the year about this time, a lot of rusts form telia. Those are the resting spores that last over the winter or through some adverse conditions. And they germinate at some point to make basidia, which is the last one. And those are very short-lived. So the telia germinate, make a little hypha, a little bit of fungal mycelium with a basidia spore at the end, which goes and infects something else and makes new pycnia. Quite a lot of rusts have alternation between 2 pretty unrelated hosts, unrelated in taxonomic sense, although they grow in the same place. So one that always I find really wacky is ferns and conifers. I don't really expect ferns to have a rust in the first place, but they do, and they're quite exciting, some of them. And a lot of them alternate with conifers. It's a really common rust on colt's foot, on Tussilago, and on some other things, and that alternates with a conifer as well. And then there's one on Arum maculatum that alternates with Phalaris arundinacea. So there are really quite a lot of wacky species pairs out there, and quite how all that evolved is still quite an interesting question. Let's not try and go there. But not all species have that alternation. There are some species that go through all of those stages on one host, and there are some species that don't have all of those stages. They might have only one of them. There are quite a lot of rusts that only have telia, for example, or they might only have Uridinia and telia and cycle, or there's some that only have Uridinia and nobody's found the telia yet. So those are called microforms. They have reduced life cycles effectively. Here is the Coleosporium tussilaginis, which is the really common one on colt's foot, but I didn't have a photo of colt's foot, so there it is on the right-hand side on Euphrasias. So if you can see the little orange bits at the tips of the leaves of the Euphrasia, which are your rust. And that alternates with Pinus nigra. So these bits here are the rust on Pinus nigra, and you can see the same just growing there. And I thought that was weird, but there we are. So all right, I've started going out there looking for my rusts. What am I going to do? So some species are really hard to find or to record. Some are really genuinely uncommon, like that one on the right-hand side. And this is also a Debbie Evans special from North Wales. So no records of Puccinia oxyrii or Oxyria digina from North Wales for ages. So Debbie was determined to go and find it she did. And there it is, with its 2 little pustules of rust sat in the corner. So that was a big search in, forgotten the name of the Coombe on the side of Snowdon. Some of them are really inconspicuous, so that's pretty inconspicuous. You have to look hard for that. That's why you want your hands and knees job. And sometimes at low density, which this one also comes at. Sometimes they're high density, but quite a lot of rusts, you have to look to find a few pustules somewhere. And then maybe sometimes there's conditions are perfect and you get a lot of it. There are some taxonomically challenging ones. There are always groups like that. So botanists have Taraxacum and Rubus and Euphrasia and all of those things. And rust people have Melampsaurus on willows and groups of rusts on grasses, which tend to be a bit messy. So there's always going to be some messy ones, but there are some that are easy as well. Tenacity was my old school motto. So it's one of those things, if you begin to get hooked in these things, you have to sort of put a bit of effort in, and then you get rewarded, and it builds on itself. So you have to have this mental checklist that says, I've found my plant, I now need to look and see if it's got a rust hiding somewhere. Centaurea nigra, always on the back, having a look, see where there's a rust on the back. When you do that kind of diligent searching, it shows that some rare species aren't. So actually, Chrysomyxa impetrae in my old version of Recorder says RDB3, and it isn't. So I reckon that if I find that in the Outer Hebrides, and I'm prepared to sit there and search long enough, I could always find a pustule of Chrysomyxa impetrae somewhere on my plant. They're not always as pretty and nice as that one, but that's a goodie. So there's quite a lot of scope for field mycologists or field botanists who are doing a bit of mycology on the side to make significant scientific contributions. There's a lot to observe here. Well, we could say the same about botany, I expect, but that's true with these. If you get a bit serious with rusts, you end up looking at spores, just partly to be sure that what you've got really is a rust. There's a magic word that goes with rusts, and it's irrumpant. So you're looking for something that's irrumpant. So if that's my leaf surface, I want something that's making a volcano of spores out like that. And if it's not irrumpant, it's probably not a rust. Probably. So here are some different spores. I'm just trying to remember what all these are. So this is actually quite a rare rust. This is Puccinia hydrocotyles, which is on Hydrocotyle vulgaris. But I've only found that one once. And this one has 2 pores. I'm not sure how easy they are to see. That one definitely has 2. One's clearly in focus and one's a bit hiding behind the plane of focus, so it's not quite so easy. But you can see it has little projections like a hedgehog around the outside. They're acuminate. This one is Eurymyces amphilidis, which is on Anthillis. You'll be surprised to hear. And this one has 3 pores. So you can see perhaps the 3 pores on each one of those. The one in the top right over there is Puccinia graminis, very common rust on some grasses. This one's a bit different in that it has a band of pores across the middle of the cell. You see that? There should be 4 pores together in a band across the middle. And the one on the bottom right is Chrysomyxa impetrae, the one we just saw on Impetrum, and that has no pores at all and has this wrinkled skin instead of the little accumulations on the outside. So those are different. They're all Uridinia, so that's all stage 2, different ones of those on different hosts. It's the Telia, the stage 3, that actually tells you which genus you're in. And there are not many genera amongst rusts. So there's the ones like this one, which have single-celled telia, and that one's Eurymyces rumicis, which is on Puccinia acetosa, and I get that right? No, I didn't. If it's Eurymyces rumicis, it's on Rumex. Yeah, thank you. So that's single-celled telia, but that's got a thicker cell wall than those Uridinia we looked at just now. Eurymyces is a big genus. Puccinia is an even bigger one, and Puccinias have these telia with 2 cells. So this one and the one up in the top right there are both Puccinias. This one's Puccinia obscura, which alternates between bellis and luzula. So the telia are on luzula. The one in the top right is Puccinia knieki, which is on, who knows what knikus is? Thistle. Very good. I'm impressed now. I didn't know that until I wondered why on earth it was called Puccinia knieki. There we are. Yeah, it's on Cirsium vulgari. And that's one of those microforms that goes through all the stages on Cirsium vulgari. So it doesn't have an alternate host. But there are some ones that are a bit different. The one on the bottom right is Phragmidium violaceum, which is a very common rust. It's the commonest fungus recorded on any foray from the British Mycological Society. So that's the bramble rust. It's the one you find always on brambles. There is another species of bramble rust. So I'm afraid you can't just look at your bramble rust and say it's that one, because there's one that has more cells in the spore with 6 or 7 separate cells instead of the 3 or 4 that we've got here. But they're all quite different. Some of them are quite pretty, like those. I mentioned Wales. So Wales has done really well recently with parasitic microfungi. So there's a loose network of field mycologists and botanists and those people who get hooked with these things, interested in rusts originally, and that's the Welsh Parasitic Microfungus group. So that started with several of us in different places doing individual field recording, and then some informal links, and solidified with putting together all of those rust records to make a census catalogue and a red data list assessment, which as far as we know is the first ever red data list for rusts anywhere. That was helped a bit as well by the Wellbeing of Future Generations Act in Wales, which says that we ought to manage biodiversity for future generations. So there was general support inside Wales for doing those kinds of things. And it ended up with this nice publication here. And I just have to say, Restr ddata coch, Fangcawod goch, a catalog cyfrifiad ar gyfer Cymru, just to prove that I can do it. So the Welsh Parasitic Microfungus group actually is distributed like that across Wales, broadly speaking. So there's Arthur Chater in Aberystwyth, there's Ray Woods just a bit further to the right, there's Nigel Stringer, who's the green dot at the bottom. The yellow dot is where I used to live before I moved to Southampton. And in the north, there's Debbie Evans, who does all those bits. So we're really quite well spread across all of those bits. And there's been especially good coverage of rusts in Carmarthenshire and Cardiganshire, with Nigel and Arthur doing those things. And Wales has a wide range of interesting habitats. You kind of know that already. It's a great place, isn't it? So wanted to make a red list for rusts, and red list criteria are not really designed for fungi because they're based on the number of individuals. So how many individuals are there on a leaf that's infected with a rust? Yeah, right, let's not go there. So let's take a mature individual to be a single infected plant, and then we kind of know where we are. And then the other red list criteria are all based on decline, but actually the recording for rusts has not been very good or systematic until the Welsh Parasitic Microfungus group got going. So we've approximated that by the extent of a fungus instead. It turns out that red listing is only really possible for rusts and smuts, where our knowledge of the distributions is okay. And the other groups that I'm going to talk about later, we've managed to census catalogue, but not managed to do the red listing because there just isn't enough good information. This is one of those gratuitous nice pictures, but this is Puccinia bistorti, which alternates between Persicaria bistorta and Conopodium in this case. It has another alternate host as well, which is another umbellifer, and I've forgotten what it is, but this is one that I know about. So that's the Conopodium and the bistort together, both infected by the rust at the same time. So you can see the kind of patches on the bistort leaves. I probably can't point out the
You have to use the pointer device.
Pointer device.
The device here.
The magic thing, is there?
I think if you press the right button.
Top one. That one?
Just make sure it's not on the screen.
Yeah. No, never mind. There's a Conopodium in there with an infection on it. So let's take it then. Conservation of rusts and smuts is kind of interesting as well. So the first step, obviously, in conservation is he's making it work. Is no, what have you done?
Go back to manual control.
Yes, there we are. So the first step in conservation is knowing what you've got. So we've got these nice assessments for Wales for rusts and smuts. In fact, there is statutory protection for one smut species in Wales already. And that's the only one that I know about anywhere as well. And so Eurocystis colchicae, which is on Colchicum autumnale, oddly enough, is protected by law in Wales. There are some special interest groups within the IUCN for rusts and smuts and for kind of everything else that's a bit random. We'll talk about some of those in a minute. But they're more focused at the moment on doing red listing activities for individual species than they are on practical conservation. So there's interesting questions about whether you should conserve all of these things that are parasites and are going to do for your plants. But I'm of course going to say that we should do all those things. So there's a lot of those kinds of things to think about. Plant life is the main interest group in the UK for conserving these kinds of lower plants. I can't say lower plants, fungi, I should have to say. I did say I'd get you a talk through the different kinds of parasitic microfungi. So the Welsh parasitic microfungus group's produced 5 publications so far. So we've already gone through rusts a little bit, Uriginales, 232 taxa in those, about 400 in Britain that I can think of. So you get a feel for kind of what you're letting yourself in for by having a look. We did a second red data list for smuts. Be more about smuts later in the talk. 78 taxa for whales. Smuts is a kind of catch-all term for a group that's polyphyletic, which means they evolve differently in different places. So there's a group of anthosmuts, and those are taxonomically the same group as rusts. They're not a smut at all in that sense. Most of the others are in the Astilaginales, and then there are these extra groups, the Exobasidiales and the Enterrhizales, which are different. So, but for the sake of what we're doing today, they're all smuts. We did an ID guide and census catalogue for powdery mildews, 127 taxa in Wales, another one for downy mildews and white bistre rusts, 132 taxa, and one for white molds. So let's talk you through some of those. Here's some powdery mildews. So there's one on Stachys palustris. That one is from the Outer Hebrides. You can see these big white patches on the leaves, which is all the mildew stuff. The one on the right is the equivalent of the telia. So we don't have telia in powdery mildews, but we have a fruiting body that forms and rests over the winter. And this is one of those, and it looks like a Sputnik. I've actually the round bit at the end is normally intact, but I've poked this one so that it's broken, and the bit that's poking out is the ascospores. So this is an ascomycete fungus. You know about those different things. All of the rusts and the smuts are basidiomycetes, which is a different group completely. But the number of ascospores is a really good character for powdery mildew. So you might be spending a bit of time poking about under a microscope to see what we've got. Downy mildews. Here's an interesting quote from the 19th century. Get a pin or a sharply pointed knife and put an atom in a drop of water on a glass for examination under the microscope. And if you see no beautiful plants there, as well developed as an oak tree and as perfect in structure as the oak, bless your stupidity, you've been born in vain. So they really are quite pretty. So here is Paraperonospora leptosperma on Triplerisperma maritimum. It's long names all round, isn't it? But if you grab a bit of this and stick it under the microscope, you see this fantastic tree-shaped bit with these. The pointy ends sometimes make really pretty bits as well. They're like a swollen bit with points on, and the spores are on the points. So there's just a couple of spores in that photo that are left. But you get an idea that there's this tree thing going on inside there. So these downy mildews, really quite interesting little group of things. And they make some of the plants they grow on grow differently. So this one on Veronica species, Peronospora grisea, makes the Veronica grow up tall. And it looks weird. So if you're out there in the field and you're wandering along and you think, that's a weird looking one of those, go and turn it over and see what's on the other side, because that's a really good clue. White molds or ramularias are not well covered by general books on parasitic microfungi, but there is a recent monograph if you really want it. It's in 3 volumes. There's quite a lot of it. You have to be a little bit dedicated, mad. One of those words, if you want to go and look at those. These are a bit superficially like the powdery mildews, but the powdery mildew has a mycelial mat that grows on top of the leaf, and the ramularias don't. Their mycelium's inside the leaf, and it's just the spores that poke out through the stomata. So you can see one there, Ramularia lamii on Lamium galliobdolon. So there's quite a lot of those which are kind of like that, and they're a bit evanescent. They kind of not much to them except the spores. This is a time to talk about plants that have dark spots on. Lots and lots of plants have dark spots, and it's quite often really, really hard to work out what is causing a dark spot. Quite often it is a ramularia. So this time Ramularia carniola on Scrophularia auriculata, but quite often it isn't. And for actually finding the spores can be a real challenge. we discovered when we were doing the field work for Ramularias that if you grab a leaf like this that has dark spots but no spores, stick it somewhere warm in a polythene bag for a day and then come back, quite often the spores are there, they've come out. So incubating can be really helpful for Ramularias in particular. But trying to identify the causes of spots on leaves is a challenge, hence irrumpant for the rusts. Remember that word, that's word for today. You've probably seen this one, have you? Yeah, there we go. This is a Taphrina. So this is Taphrina olney, but there are several other Taphrinas that cause bumps in leaves. There's a really nice one, which I could have put a photo of in, but I didn't, which makes the catkins of Populus go bright yellow, which is lovely. So there are quite a lot of Taphrinas that you could look at. In fact, the Welsh fungus microcytic, whatever it is, group is currently looking at Taphrinas and chytrids for the next book in the series. So we're doing quite a lot of work with those. Taphrina is nice because they're kind of big and you can see them. Chytrids are quite nice, but they're not very common. Syncytrium you might have seen on dandelions. There's a Syncytrium taraxicae, which makes dandelions go all warty with yellow lumps on. And there's a Physoderma, which I'll show you in a minute. As well as those, then there's a whole load of parasitic ascomycetes, some of which are rather tiny and inconspicuous. And then there are deuteromycota, deuteromycetes, which is a group of fungi that don't actually have a perfect stage, and it's a bit difficult. They're kind of just a bit of stuff that grows on your leaf. So there are plenty of fungi out there. Chytrids I'm going to mention. So I got excited a little bit about chytrids because I could find some of those. So up in the northwest of Scotland in particular, Syncytrium succisii on Succiser pratensis. But how much Succiser pratensis must there be around the country? Do you ever see those? I see them in Scotland, but I don't see them anywhere else. So it may be that it is a bit Scottish, but it's not everywhere. So the one on the right there is Syncytrium stellarii, second record for Britain, as far as I know, on Stellaria pallida, and that one's from Hampshire. So there are things that are found in places that are not Scotland, honest. There do, on the other hand, seem to be more parasites in the west and the north, so where it's wet. I think that the wetness really does have an influence, so people in the south and the east might have more challenges. This is one I got excited by. So this one's called Physoderma potteri. It was described from Northumberland in 1926. When I did a bit of digging, it was known from 2 sites in Northumberland and 3 in New Zealand. Why they're as far apart as you can get, more or less. I'm kind of guessing the New Zealand one might be introduced, but I don't know that. More recently, there are some records from Germany and Italy, but then it turned up in 2011 in the Outer Hebrides. I got one thinking, oh, that's a gall. It's a round gall growing on a little thing. There must be a maggot fly or something inside. It'll be an insect, and it wasn't. It had spores inside. And since then, I found it in several more places in the Outer Hebrides. So it's not common, but it's not exactly as rare as it looked either. But as far as I can tell, those kind of 10 records there are the only known places in the world for it. But is that just because people don't look? It's not as conspicuous as you think. It's quite well hidden. So that's one to look for if you've got lotus. Everybody must have lotus, mustn't they? Because the Welsh Parasitic Microfungus group has put in quite a lot of effort, we've discovered that things are much commoner than we expected. So we started off with reasonable data on rusts, but each time we've done another group, then we go out in the field and try and find stuff. And over there is Entyloma hellocyadii on Oenanthe crocata, also occurs on some other similar dear. My brain is not functioning. Umbellifus. Thank you. That's the word that I'm looking for that isn't coming out. It's also on Apium nodiflorum, for example. So that was new for Wales in 2011, but actually it turned out to be more or less common everywhere. So if you go and look for it, you can find it. And I think that's true for quite a lot of these kinds of things. They're not recorded just because people haven't looked, not because they're not there. And it's I need to get a kind of search image for the thing that I'm looking at. And once you know what it is that you're looking for, then it turns out to be everywhere. Doesn't that happen with plants as well? I don't know what one of those looks like, but I found one day I'm lucky and I find it, and then the next day I find it again, and the next week is one more, and on and on. So we did big efforts with Ramularia, and as I say, currently on Taphrina and chytrids, sometimes easier and sometimes harder. I promised you some taxonomy, not very much, honest. Identifying these fungi can be a bit challenging sometimes because the fungi are quite reduced. There's often really only spores, not much else to make an identification from. Sometimes you get a few other bits like mycelium. But I said they were easier than mushroom-shaped things because you can see what it is that they're parasitizing and identify it. So that's a really big aid. And there have been two schools of taxonomy. Britain started out being the hotbed of rust research. So there was Plowright in the late 19th century who did lots of stuff. But we kind of lost that to the Europeans coming in a mo. Later on, then Henderson came back and worked particularly with sedge rusts in the 1950s and 60s. And there's another guy in Scotland in the 1940s who did those kinds of things, but they're coming along. And they largely lumped stuff together. So they were in the lumping kind of category. And the continental workers, the Germans, the Swedes, and the Swiss, did a lot of experimental inoculation. And that's been followed up by a lot of DNA work in more recent times, especially in Germany. And that's tended to divide things back up again. So although when things got lumped in Britain, the Europeans carried on having things separate. So that's given us a more divided taxonomy, but it's actually even easier on the plant hosts, because if you know it's this host, it's got to be that species. So we've got a lot of these narrowly host-specific species, and DNA results suggest that there are lots of those. So different host means a different parasite. Occasionally there's the same parasite on multiple hosts, but usually closely related ones. That means your older records can be a bit hard to assign, but if there is an identified host or the specimens there, then you can do something with it. The Welsh Parasitic Microfungus group has been using taxonomy from the Europeans, which is in this fantastic book, Pflanzen Parasitische Kleinpilze. I didn't look to see if there was one in the Summerfield books, but if you read German and you're interested, this is the one to have. It's got rusts and smuts and everything I've talked about except the Ramularias. The Ramularias are not in this one. The Welsh red data lists and ID guides provide ID help, which is basically taken from the kind of taxonomy that's here. It's the ID guides for Wales are poorest for rusts because we started with that one and they're kind of well known, but they're quite good for the other things. And we're working on a hand list which will bring all of that local nomenclature in line with the continentals and make an accessible ID guide. So that's still to come. And then just towards the end, I'd like to say, I said I got a bit hooked here. So I'm got a bit obsessed with smuts, and this is how it happened for me. If you go this way, you'll no doubt it will be different for you. So after I got encouraged, rusts seemed like a good extra activity for botanizing in the Outer Hebrides. Should explain that I can sometimes go out in my Vice County, I can stroll, yomp into a tetrad, can spend all day in it, and I can come out at the end with just over 60 species. So I decided that finding a rust while I was doing that might be some really nice extra activity. So they're eminently recordable, and I do go out and record them. Smuts are a bit more challenging. You often find only 2 or 3 in a day if you're lucky. But they're still an extra thing, so something to get excited about. So I do get excited when you start looking. It doesn't take you long to find things that are interesting. How often does that happen too? So I started out with an early spring visit, and I found spring squill, Scylla verna, and it has this. This is one of these anther smuts. There it is, all the anthers making this black powdery stuff. So found that one. I looked it up. It was an aggregate name. But I'd done a bit of reading. I knew some people, so I contacted a guy called Matthias Lutz, who's in Tübingen, and I sent him a specimen, and he did the DNA for that one. And it turns out to be one of those narrowly host-limited species, a new one. So here we go, Anthrospora tractimi, because Scilla has been split up, at least in some people's taxonomies. So it's now Tractima verna. And Arthur Chater had that one in Cardiganshire as well. Result was scientific paper, new species. Very good. Now I'm really interested. What else can I do? What else can I find? So wander around my Vice County. Here's another nice one. So the doesn't photograph very well, but this is Anthracoidea scurpi. So those black bits on the photo on the right are little balls of smut spores in the top of replacing the inflorescence in Trichophorum. This is actually Trichophorum hybrid, so it's Trichophorum forsteri, and it seems that these things preferentially infect hybrids. So actually they're quite a good taxonomic clue as well. I have found it on the species since, but quite a lot of the infected stuff is hybrid. On one particular visit to North Lewis, it turned out to be frequent on the moorland, but there were not many records for Britain, and I wrote a little article about those for Field Mycology, which is a nice little journal. It turns out, now that I know what I'm looking for and I do this regularly, that this smut has occasional boom years. So sometimes I find it everywhere I look, not lots of it everywhere, but I mean that it's all over the place. Some years I don't find it at all, or I find one infected spike, not even an infected plant. So it seems some years are good and some years are bad, and that's just how it goes. And I think that's quite true for quite a lot of these parasitic fungi. But Trichophorum's really common on moorland. I mean, how much Trichophorum is there in Scotland? So this must be more places than we know about. So that was another exciting one. That's 2 down. Now what shall I do? So then I was out in one of my horrible tetrads. This was when I did it, it was the most species poor complete tetrad that I'd found, with 63 species in. And during the process, I collected a manky leaf of Glyceria fluitans, thinking that might have a rust on it. It's a bit manky. I'll take it home and have a look. And it didn't have a rust. There was nothing irrumpant. But it did have a smut. And this is a really weird smut. It's got spores embedded in the leaf tissue, and it doesn't do anything much on top. So you don't really see it. But I had to dig into the tissue to see what was there. And there were my nice thick-walled spores under the microscope. And that was Ustilentaloma fluitans, new for Britain. So there we go. That's another short note. So we did the DNA with that one, wrote that one up. That was lovely. It's a bit completely unphotogenic, so I can't give you a photo. Relatively few records anywhere, but that's probably just because people don't look for it. And I have found it again since. Now that I've got a better idea what I'm going to look for. I said that the west was often better and the north is better when it's wet and windy. I think that's good conditions for infection, and there are relatively lots of species and specimens. It may not be quite so easy in the south and east. This is another spring rust. This is a weird one because these are the Telia. Telia don't usually happen in the spring, they usually happen at the end of the year. But for some reason, this one, which alternates with Sorbus, produces Telia in the winter and then Uridia in the summer. And it has these, when it's wet and rains, the Telia swell and make these orange things on the twigs of your juniper bush. It looks really weird. This is one of the weirdest rusts I know. There are some southern ones as well. There's one that grows on Juniperus chinensis, which is not the same species, different gymnosporangium, alternates with pears. Got a pear tree?
Yes, Cambridge was relatively infrequent. And Chris Preston wrote a paper on it about 4 years ago in our local natural history journal. We had a gall finding visit with our local natural history society a couple of weeks ago. Thought, oh, there's a pear. I wonder if it's got this thing on it. Oh, wow, this is really exciting. It had, and the next 3 pairs that I looked at in different tetras also had it on. So every single pear tree that I've looked at in Cambridge has this pear rust.
John's well on the way to being a converted mycologist here, so there we go. Nowadays, I live on the south coast, and my daughter takes sailing lessons. So on a Saturday morning during the summer, she's off doing those things. And one day, my wife rang me up from there. Sometimes I go and watch, sometimes I don't. I have to do the shopping on Saturdays. So one day I rang up and said, it's a nice day, come and see what's going on, and we'll have a walk afterwards. So we walked along the front, along the seafront by the sailing club. And I can't go for a walk without looking to see what's there. So there was another of those funny looking insect galls on Silene uniflora. But when I opened it up, of course, there wasn't an insect inside, there were spores instead. So this one is, it's a smut, Thecaphora melandrii, also new to Britain, just like that. Thecaphora melandrii has multiple hosts, so it's got at least 3 different hosts. One in Stellaria, or Stellaria graminia is another host. But we checked the DNA for all of those; they're all the same, and then did a little bit of research to work out its world distribution. And as far as I know, that is its world distribution. I tried really hard and went searching for a lot of stuff. All the yellow things are old records, and all the green things are new ones. So people are not looking very hard for these things currently, as far as I can tell. So it took quite a lot of effort to get me a map like that, but that's really quite interesting. And you can see the different hosts because they're coded with different shapes. And that green triangle there is my record. Turns out to be, having found it near the sailing club, it's also just down the road from my house. But I didn't know that, and now I know what I'm looking for, and there it is. Also got excited, this is a bit esoteric, but there's a series of smuts called enterrhizas, which grow on the roots of Carex and Juncus. So yeah, you have to be a bit dedicated to go around digging up all of these things and see, but all right, I got interested in some other things. Let's try one of these. So pretty much the first Juncus bufonius I pull up in the Outer Hebrides, smut on the roots. Fantastic. And it turns out that quite a lot of them are quite common. Arthur Chater got very enthusiastic about these and searched for different species. And they turn out to be frequent, not just in the north and west, but all over the place. And the one that I've shown you a picture of, can see the Eliogeton fluitans, which is kind of round the outside of the picture. And I pulled up a bit in the middle, and those white bits are all the root smuts, and they're floating off. And I'm pretty sure that's how they're getting dispersed as well. So there's some research to do here. Nobody really knows much about these things until Arthur started finding this particular species in Wales. It was known from one site in Denmark and nowhere else. But it turns out to be everywhere. It's all over the Outer Hebrides. It's all over Wales. If you've got this, go and look for it. It'll be there. And there are quite a lot of unresolved questions. So here's another root smut. Can you see this big swelling here with lots of little white bits? That's on Juncus bulbosus. I'm pretty sure that the one on Juncus bulbosus is different to the one on all the other Juncuses. It's never been described. Just waiting for the DNA to come back, and then probably that will be a new species as well. So there's probably a whole load of undescribed, narrowly host-specific species out there that we just don't know about. Enterrhizas are evolutionarily ancient, so they split off from the rest of fungi a long time ago, kind of pre-dinosaurs, and they've just been sat here. So it's not really clear whether they're a group that's been sat there forever as not many species and then suddenly radiated recently, or whether they radiated a long time back and then all been carrying forward. All those kinds of questions still to answer. Lots of species of Microbotrium. So those are one of those ones that are anthosmuts. So they're called smuts, but they're actually taxonomically rusts. And there's one of those on butterwort, Pinguicula vulgaris. I looked at that a couple of times in the Outer Hebrides. The truth to tell, I was too early in the year and it mostly wasn't in flower. So I never found it. And having not found it a couple of times when I looked, I sent an email to my fellow recorders saying, can you find this smut on butterwort? And the next day, Ray would send me back an email saying, have you found it yet? Rotter. And it turned out it was just down the road from his house. And it turns out to be common in Wales. And in fact, now I've gone at the right time, it's common in the Outer Hebrides as well. And lots of people are finding it in Scotland. If you've got Pinguicula vulgaris, go and look when it's in flower. You actually have to pull the plant to pieces to see. But if you do, you find this anther with a brown smutty bit at the top instead of the normal yellow pollen stuff. It's obvious that it's there. So we did a DNA on that one, a little paper again with Matthias and Arthur and Ray. And as I say here, several other people have searched. It turns out to be widespread. If you can find one in Pinguicula lusitanica, I'm really interested because nobody's found one of those yet. So I keep looking, but I haven't found it. So I've been well and truly parasitized around all these things. There's an awful lot out there that's still unknown about lots of these plant parasitic fungi. Naturalist observations endlessly valuable, so you can make a real contribution here by doing a bit of work in your local patch. And I think that's more important than ever in a world where observational research is not fashionable or funded. So it requires some application to do the background research bits, but for me at least, it's been endless hours of fun and much easier than mushroom-shaped things. And on the right there is Puccinia punctiformis 01. That's the Pycnia and the Esia on the creeping thistle, and it makes it stand up. So you'll see this one. It looks yellow and spindly and pale and upright. Yeah, there's lots of it. Yeah. So I have to do some thanks bits. So the Welsh Parasitic Microfungus Group, that's Arthur actually looking for Enterrhizobranchiariana on Eliogeton. Sadly, he wasn't with us. We got awarded a small award from the British Mycological Society for all the work we were doing with those things. So that's Ray and me and Nigel Stringer and Debbie Evans. Big thanks to Matthias Lutz in Tübingen and Martin Piatek, who's in Krakow, because they've helped a lot with the papers and the DNA and those things, and lots of people and organisations who've helped us and provided funding and done all sorts of things. A few references, just so that they're here somewhere for the things that we have done, and those are the 5 books that we've produced. Didn't check whether they're in Summerfield books now, but possibly they are. Righto, good. So if you want, those are not very expensive. The big German book really is expensive, and you have to read German. There we go.
Certainly with my own local naturalistic society, we start moving away from plants to other things at this time of year, and galls on plants is one of them. And as I say, finding some of these really quite spectacular galls on the older cones was really quite nice, because somebody had the gall book and said, oh, it'd be nice to find one of those. And the next older they looked at, there it was. So some of them are fairly easy to find. I think another point is perhaps some of them are becoming more frequent, not just because they haven't been looked for, but because they are genuinely becoming more frequent due to climate change. And that may be an issue as well, that only recording will show. Anyway, anybody got any questions?
Just an offer of help. If you need assistance with downy mildews in mathematics, I did my PhD on them, and I've actually got quite a lot of early literature still there in my cupboards if you ever need an assistance with them, because they very often get overlooked, even more so than nowadays.
Yeah, willing volunteer. Somebody needs to write a monograph of downy mildews.
Sorry?
Somebody needs to write a monograph of downy mildews.
Absolutely, because
Mark?
they've been produced over the last 20 years, and the literature's all over the place.
Yes.
Yeah.
For quite a lot of the other genera, there are monographs, but for downy mildews, there isn't. So if there's somebody who's really desperate for a project.
Is that beautiful one bottom left? Is that on bog rosemary?
Yeah.
They're the flowers that are contorted.
No, it's the leaves that are contorted.
Wow.
That's the Exobasidium. There are some of those on Vaccinium as well.
So can
When you've got something new that's likely to have multiple hosts, how the hell do you find the host? Is it you've got to guess sort of something in the vicinity and inoculate and see if it works, or
You mean the alternation bit? Yeah, the Germans did a lot of that kind of experimental work in the 1900s, the early 1900s.
Yeah.
So we kind of know for most things, but there are definitely some that's still not known. And then you would.
particularly difficult when you've got multiple hosts, because
Yeah.
you might find one and think that's it. You know?
Yeah, so you could do those. Got a greenhouse? Careful inoculation experiments? Yeah.
Alison, do you have a question? No, it's Alex. Yeah.
I was just going to ask, My go-to reference for these sorts of things is Microfungi on the plant plants, Alice and Alice.
Alice and Alice.
80s, I think. Is that just too out of date now?
It's a fantastic book, but then the nomenclature is very out of date. So if that's what you've got, then by all means use it, but the names are updated. So you ought to get one of those or a set of those at the same time, and then you can look it up in Alice and Alice and then check and see what the new name is.
Hello?
When people make gold ones, I think they're going to do gold cards. Do they get shared with my colleagues? Well, the ones that are formed by my.
Yeah, that's a good question. So I'm in both societies and I kind of do fungus calls, but not automatically, I think is the answer.
I think my impression with the pear rust was the mycological society probably has more than the fungal society. And there didn't seem to be much an overlap. So I think Chris Preston found 30 records in one database and I found 2 in the other. So
Well, there's a huge unhealthy interest in galls in Somerset.
Why is it unhealthy?
But I think the record will go to the Gall Society, but I don't know if it goes to the college.
Yeah, I think it all ends up on NBN eventually.
Right,
my impression was it didn't end up on the NBA
Either. Oh, right. Okay.
that'll be the record for Gall. The record for the fungus, but it ends up on the
I think the Tefrina wasn't on the NBN for Cambridgeshire at any rate. So I thought I found a new county record and I hadn't.
Yeah, that's a good question, and I'll ask it further up the chain for you and see what we get.
Well, thank you very much indeed. That was an absolutely fascinating insight into something that we perhaps gloss over